Thank you for joining us today. I'm Helen Ekman, a member of the Community Advisory Board for the NCI Designated Cancer Center at Sanford Burnham Prebys. I'm a stage 4 cancer survivor, thriver, and I'm married, mother of 4 with 9 grandchildren. I'm also a full professor of business at the University of Massachusetts Global. We're excited to have you with us as we talk about the amazing discoveries and collaborations that are happening here at Sanford Burnham Prebys. Today, we will unravel the genetics of congenital heart disease. Congenital heart disease is one or more problems with the heart structure that exist since birth. Some congenital heart defects might not cause any problems. Others, however, can cause life-threatening complications. While congenital heart defects remain the most common cause of death in infants, the genetic basis of these defects is unknown in many cases. Solutions are urgently needed for these most vulnerable patients. To pave the way for better care and prevention, Sanford Burnham Prebys has teamed up with Rady Children's Hospital to understand this disease by studying the genes of actual patients. This partnership ties the research community with clinicians and patients. What is learned in the lab is put into practice in the clinic. The results we are seeing in the clinic inform our research in the lab and make it better. Today, we will hear from a scientist and doctor working together, collaborating to help infants and their families impacted by congenital heart disease. And we will hear direct from a patient family that is receiving life-saving therapies in thanks to this research collaboration. We'll discuss what is being done to help researchers predict at-risk patients and improve precision medicine. And we'll cover how testing gene candidates can lead to the discovery of new genes and pathways to help these infants. Please know these discoveries are not possible without you, our viewers. You make this work happen. It is because of your support that we can provide the help and care that is so desperately needed for these families. We thank you. Before we begin, if you have any questions during or after the presentations, please use the Q&A button at the bottom of your screen to ask them your questions at any time. We will then address the questions following the presentations when we all come together to review, okay? Now let's begin with Dr. Rolf Bodner. Dr. Bodner is the director of the Center of Genetic Disorders and Aging Research and professor of Development, Aging, and Regenerative Program at Sanford Burnham Prebys. Rolf discovered the Tinman gene, which we will learn about today. He also established the fruit fly heart as a research model for cardiac development, function, and aging. The fruit fly heart model has led to a deep understanding of the molecular genetic basis of the heart, as well as how the heart is formed. Dr. Bodmer has published over 100 research papers, authored numerous book chapters, and serves on several of the scientific advisory and editorial boards. Welcome, Rolf.
Well, thank you very much, Helen, for this very kind introduction. It's really a great pleasure to tell you another story of the Wizard of Oz that you all know, which features as the main character Tin Man. So as Helen already has described to you, is that congenital heart disease is the most common human birth defect, but we know very little about the genetics that are associated with it. In many cases, it is really the abnormal gene functions that are causing, during development, that are causing this disease. So let me tell you a little bit about our approaches, how we go about to study them. And for this, because it's such a complex organ and humans are such complex organisms, we turn to a much more simpler organism, the fruit fly, that many of you don't know, have actually a heart. As you can see here, the fly embryo has 2 rows of cells that are the developing heart. So it's a very simple system, and you might not even know the fruit fly has a heart. But in one mutant, which I then call Tin Man, these embryos don't have a heart. And so we thought this could be a good entry point to understand in a very basic fashion what makes the heart. What is the complement of regulatory genes that actually makes the heart form and in the right position? And so the Tin Man gene was the first one that was discovered of this kind. Let me show you a little bit how the heart actually forms in a time-lapse movie in the next slide here. You can see here the 2 rows of cells that come together to form this primitive, or it's a simple heart tube that pumps the blood in flies. Let me show you it again. These 2 rows of cells come together to form the heart tube. So it's a very simple system. And we thought we could learn quite a bit in terms of basic mechanisms of how this works. So here is the gene expressed in these developing fly hearts that come together into this row. So how is this similar, or what can we learn for a human heart? In fact, any animal with a heart has precursor cells that are also coming from 2 sides of the embryo together to form a primitive sort of heart tube, like in your heart when you were an embryo, to then develop further and form chambers. But the very simple early steps are exactly the same. And mutations about 25 years ago discovered in the Tin Man gene in humans have then been shown to cause congenital heart defects. So we thought this was a really good entry point to understand how the heart functions, and in disease, how it malfunctions. Let me show you what the heart looks like. So here at the bottom, you see a beating heart of an adult fly that we want to study and see how it not develops properly in mutant and abnormal situations. So we would like to learn in this heart tube in the fly how the human heart will eventually be formed. And why? What are some of the advantages? Because the fly heart is a very simple system and beats on its own like the human heart without nerve stimulation. It just continues beating in a regular fashion normally. And it is composed of very similar components as the human heart. So the similar proteins that make the heart contract are also functioning in the fly heart. And most of the disease-causing genes or heart disease-causing genes, and actually almost all disease-causing genes, are conserved in the fly. So it's a very simple system that can tell us some of the basic ways by which the heart is developing and what could go wrong if it's developing improperly. So for this purpose, we have hooked up with people like Paul Grossfeld and other clinicians and human cardiologists who study patients. And in fact, these days, many patients that come into the hospital, their genome is being sequenced for potential damaging mutations. And so we get this information from these hospitals, and we then test them in our model system. We test them in cells, so human stem cells that have been formed into heart cells, but also model systems like zebrafish and flies. And I will tell you the story how we do this in flies. And I hope you can appreciate the power of this kind of approach. So with this information of the genes that could potentially be affecting heart development and could go wrong in patients with congenital heart disease, if we identify what they do in our model systems, we can put them together into a network. And we can do that because we can study not just one gene at a time, but several genes at a time to see how they in fact interact with one another. You know, what is dependent on the other. So that generates then this hairball, so to speak, but it's very useful to then understand what are the connections, what are the interactions between the genes. And that's then very useful information going back to the clinic to understand what could be the cause in each individual patients that for the heart do not develop and function properly. We have done such an effort actually in high throughput in a few years back with the Mayo Clinic, where we tested 1,000 genes that they had bioinformatically prioritized as being potentially contributing to the disease of heart disease in about 130 patients. And we're still continuing this research to identify what could be the complement of genes malfunctioning in each individual patient. So this is going to be a long-term project, but I think we have some good angles to it that are already showing fruits in that direction. So how do we do this in flies? I hope you can all see this movie here, where we have glued down the flies onto their backs, where the heart is, and you could see the wiggling feet here, and we image them the heart that has been fluorescently marked with high-powered microscopes. And this is then what we look like. This is what we film the heart. We can have in great detail, we can see how the heart contracts, how well it contracts, how fast it contracts, how rhythmical it contracts, and so forth. And though any abnormalities that we see, we can identify. So as you know from an electrocardiogram or a sonogram, if one takes a cross-section and lines up these contractions in time, this looks like what the clinicians call an M-mode. And we can then automatically determine all the parameters of this heart. And for example, I show you here an abnormal heart that has abnormal contractile proteins. And you can see this heart doesn't contract very well. It's dilated as if it was dilated cardiomyopathy. And so we can identify many phenotypes in the fly heart. So importantly, we can then study not just one gene, but several genes. And you see that, okay. So this is the normally contracting heart. If we have a Tin Man, a weak mutation in Tin Man, and maybe another gene, that could cause maybe not much defect. This heart contracts almost the same way and forms, and is morphologically formed the same way. Another gene, if you put it together with mutants of Tin Man, has a different effect. As you can see, it's already deformed here, and this portion doesn't even contract properly, and it's thinner. And when we now put all three genes together, we can see really the power of how one can do combinatorial genetics, as we say, or polygenic assessment of heart function. And you can see that heart now has developed into very thin heart. It has not really developed much at all. It hardly contracts. And so very dysfunctional. Why this fly lives, you might ask, is because in the fly, oxygen is carried by a tracheal system, not by the heart. So they can still live a little bit, but they will die prematurely. So I hope I've convinced you that we can do this combinatorial genetics to identify which genes are working together, and then have a whole network of genes for congenital heart disease, how they could be working together. Because it's, of course, not just one gene that acts in isolation. It's many genes have to function in a very coordinated fashion. And so this information, we bring then back to the clinic for them, for the clinician in the first pass to have diagnostic information, which combination of genes could be important in their disease, and then also to find avenues for therapeutic interventions of many different kinds. And you will hear this much more from Paul Grossfeld in his talk. And so I hope I have convinced you or made you think about when you swat the next fly that they could actually be very useful in translating scientific discoveries to improve potentially patient care. And Paul will tell you an example of how identification of Tin Man in the fruit fly has led to life-saving interventions in patients with congenital heart disease. So I thank you all, and I'm awaiting your questions at the end of the presentations. But let me thank you also, the Wizard of Oz, who has enabled Tin Man to find his heart. Thank you.
Thank you very much. It's encouraging to hear that Tin Man has found his heart. I think it's fascinating that you are working with fruit flies and you are making these amazing scientific discoveries. And thank you for sharing that with us today. I was on the edge of my seat. So now we look forward to hearing from you later on in the program, and that you'll be available to answer some questions then. So thank you. So next, sorry. Oh, you're so welcome, and thank you. So our next, we have Dr. Paul Grossfeld, is a board-certified pediatric cardiologist at Rady's Children's Hospital, San Diego, and an associate clinical professor at UC San Diego School of Medicine. He treats both pediatric and adult patients with congenital heart disease. Dr. Grossfeld has a particular interest in treating patients with rare genetic syndromes and serves as the chief medical advisor for the 11Q Resource Group. He also directs a basic science research laboratory at UC San Diego dedicated to studying genetic causes of congenital heart disease. Along with his clinical and research responsibility, Paul directs the Pediatric Cardiology Fellowship Program and is actively engaged in teaching fellows, residents, and medical students. Among his professional activities, he and his wife, Susan, lead an annual cardiac surgical mission to Angkor Hospital for Children in Siem Reap, Cambodia. Dr. Grossfeld also serves as a cardiology consultant for the United States men's and women volleyball teams. Welcome, Paul, and thank you for the good work you're doing.
Thank you, Helen, and a huge thanks to everyone at SBP who put in really a lot of work to make this happen today, and to everyone who's attending. I really appreciate that. It's a tough act to follow to come after Ralph Bodmer, who is one of the most unassuming yet brilliant scientists. And I want to really commend everyone from Ralph's group, because unlike a lot of really outstanding scientists who are always trying to do the right thing, but Ralph and his group are really exceptional in their passion, as you're hearing today, for translating what they learn in the fruit fly to helping human patients. And my approach, as you'll hear in a minute here, along with Ralph, is to actually start with a human patient and hopefully identify a gene or the genes that are responsible for particular heart defects, and then utilize the incredibly powerful genetic tools that we have available to us, like in the fruit fly system, to then learn as much as we can about these genes, how they function in normal heart development, how their loss of function can result in specific problems with the development of the heart, leading to these congenital heart defects. And ultimately, as you'll hear in a couple minutes, apply that knowledge to hopefully helping to improve patient care that, in this case that you're going to hear, can actually result in life-saving interventions. And our ultimate goal is to really put out of business my dear friends, the surgeons at Rady's, that will hopefully someday allow us to prevent these heart defects. So as Ralph said, they discovered the Tin Man gene, and I want to share with you a really interesting story about how that gene has helped us to advance patient care. Now, for some reason, my slide is not advancing. There we go. Okay. So I want to introduce you to a family that you're going to meet live in just a couple of minutes. This is the Mitchell family. And so this is Melissa, the mom, and John. So Melissa and her two children, Chance and Cadence, were all born with congenital heart defects. And this is just the way a geneticist draws that. Melissa was born with a hole in her heart, and subsequently not only had surgery to fix that hole, but years later developed problems with the electrical system of the heart, what we call an arrhythmia. Chance was born with the same type of heart defect as Melissa. And up until I became involved, no one suspected any problem with his electrical system. And then I became introduced to the Mitchell family when Melissa was pregnant with Cadence, who was born with a much more complex heart defect, something called hypoplastic left heart syndrome. And so based on this constellation of heart defects in this family, I recognize that this family may actually carry a mutation in exactly that Tin Man gene that Rolf has described for you. And so we have the power to do whole genome sequencing, or what we call targeted sequencing, to specifically analyze in the human patients any gene of choice. And lo and behold, we found that Melissa and the two children have a mutation in the Tin Man gene. So that was very satisfying in many ways. One, we found the cause, so that's great. But it also raised some interesting and very important clinical issues that could directly impact our plan for how to manage these patients. And so if you look at this tracing here, this is what's called an EKG, or a rhythm tracing, and each one of these vertical lines represents the beating heart. So normally a heart beats at a regular interval like that. And in this case, what we discovered based on our understanding of what the Tin Man gene does is that this electrical system already, this was from Chance when he was only a few years old, was already beginning to show signs that it was failing. And we knew that this can be a progressive progress that could ultimately, God forbid, result in the heart stopping completely, what we call complete heart block. And so because of that information, when both Cadence and Chance had their heart surgeries, we elected to put in what turned out to be a life-saving pacemaker as a backup system to be able to compensate for this kind of dysfunction in the electrical system. And at that same time, Melissa knew that she might have had some kind of arrhythmia. She was still running marathons at the time. And I immediately suggested that she follow up with her adult cardiologist to do a more systematic analysis. And it turned out she had an even more advanced problem with the conduction system of her heart, such that it actually put her at risk for sudden cardiac death. And in fact, that's how this gene was originally discovered in human patients that not only had surgery for a hole in the heart, but years later were suddenly and completely unexpectedly dying, because up until then, nobody knew about this potential comorbidity associated with this gene defect. So as a result, all three family members have received life-saving pacemakers. And that was actually a number of years ago. So now, as we continue to follow Melissa and the two kids, Mom now has absolutely lost all of the intrinsic spontaneous electrical activity of her heart, meaning she is 100% dependent on that pacemaker, which in real terms means that had we not put in that pacemaker, she wouldn't be alive today. And both children have also shown progression, ultimately probably towards that same fate, such that without the pacemakers, they would be at great risk for sudden cardiac death. So here they are. This is years later. We fixed them. And thankfully, because of our knowledge that all started with the work in Rolf and his group, with an index of suspicion, we could now intervene in a very significant way. So what I want to do is switch gears and tell you about a different story that has continued to keep us, our groups, working together, about a new gene that my lab discovered many years ago. And this story actually started back in my training at UCSD in pediatric cardiology with this little boy who I was taking care of because he had one of the most severe congenital heart defects called hypoplastic left heart syndrome. And I think you can appreciate here, he has some other problems. And this is a picture of his chromosomes. And if you look at this, what's called a karyotype compared to his normal 11th chromosome, I hope you can appreciate here that he was actually missing a chunk of that chromosome. And so we postulated that there must be one or more genes in that region that likely is the cause of this child's problems, including hypoplastic left heart. And in fact, this region contains probably somewhere around 100 genes, and we were ultimately able to take advantage of our technologies to pinpoint the gene to one that's called ETS1, or in the fruit fly, it's called pointed, as the cause of heart defects in this rare syndrome called Jacobson syndrome, and also likely in some subset of patients in the general population that also have these kinds of heart defects. So to give you a little bit of background about this particular heart defect, so this is just a cartoon of a normal heart on the left. So this is the left ventricle, which is the main pumping chamber, which pumps blood out to the body by way of the aorta. And there are two valves, the mitral valve and the aortic valve, which normally allow blood to flow forward without leaking backward. In hypoplastic left heart syndrome, what happens is this left ventricular chamber is very abnormally formed. So it's too small to be able to pump the amount of blood required to maintain the needs of the body. Going along with this so-called hypoplasia of the left ventricle is a very diminutive, sometimes completely blocked off mitral valve and aortic valve. And compared to the normal size aorta, the aorta in hypoplastic left heart syndrome can look like just a little string. So this is a uniformly fatal congenital heart defect that accounts for about 3% of all children born with congenital heart disease. So in the United States, there's somewhere between 1,000 and 2,000 babies born per year. And the costs directly are probably in excess of $1 billion a year. Now, if you look at this cartoon, it interestingly shows something that I'm going to focus on in my next few slides. And compared to this normal left ventricle with a relatively thin wall, what you see here is this massively thickened wall, suggesting that the diminution of this chamber may be due to an inward growth of that wall, of that tissue, as I've shown here with these arrows. So with that in mind, we wanted to take advantage of some of these very powerful yet simpler genetic systems, like what you heard Rolf talk about. This is actually the heart of a frog. And what you see here is a normal ventricle, and we can use our genetic tools to specifically knock out the function of that same gene in the frog. And lo and behold, what we see is something reminiscent of what I just tried to lay out for you. Specifically, there's this now abundance of heart cells that impinges on that chamber to cause what effectively is a hypoplastic left heart in a frog. And just as you've already become experts now in the fruit fly heart, if we look at what happens when we have a mutation in this same gene in the fruit fly, compared to the normal fruit fly embryonic heart, we see this same concept of an abundance of cells that are basically clogging up that chamber and impeding its ability to pump the fruit fly's version of blood out to the body. Now, coming back to the frog, what we've actually already been able to demonstrate is potentially with very early intervention as this heart is developing, we actually may be able to prevent that. And that would be a totally game-changing approach in the field of congenital heart disease and pediatric cardiology. Now, interestingly, there aren't a whole lot of genes that are involved in heart development. Yes, there are hundreds, but it's limited. And remarkably, these same genes that I've told you that are in the human genome are in the fly genome. And so similar to what Rolf showed you before, we wanted to see if there's any sort of relationship in the function between the Tin Man gene that Rolf had identified and this pointed gene that we had identified from our human patient. So I'm just going to show you a couple of movies of basically what looks like a normal heart. This is actually the heart of a fruit fly that is a carrier of the Tin Man mutant. And this is similarly the heart of a fruit fly that is a carrier of the pointed mutant. So they both look like they're looking pretty good. But yet, if you combine them together, what you see, hopefully this will project, is a much smaller heart. And graphically, we can show here that compared to those other three hearts, when you have the two mutations from the two genes together, you get this dramatically decreased heart function or cardiac output. And what that tells us is, remarkably, it was meant to be for Rolf's group and mine to work together, because it just so happens that our two genes do indeed appear to function together in a critically important, highly conserved genetic pathway involved in heart development. So we have embarked on a lot of different approaches to identifying genes. And hopefully what I've, along with Rolf, shown you is that once we can identify these genes, it allows us to make diagnoses earlier. And as we learn more about the function of the genes, we think that not only can it help to improve in our management of these patients, but we hope in the future someday to be able to apply that knowledge to prevent these very devastating congenital heart defects. So to kind of put all this in perspective, this young lady is actually the first baby that I ever diagnosed with hypoplastic left heart syndrome. It was actually about 25 years ago now. She's undergone 3 open heart surgeries to try to salvage the good part of her heart to do the work that the left part of the heart was supposed to do. She's also had multiple cardiac catheterizations. She's on multiple cardiac medications. And as you can see there, she's a beautiful, highly intelligent, now 25-year-old. But we don't know what the future holds for her. And so that's why it's so important for us to learn more about the genetic basis and mechanisms of these heart defects. So hopefully she will, like every other child is supposed to do, outlive her mom. So again, I want to thank all of you. One take-home message I think I want to impress on everyone is actually what's shown here is that to really take on these kinds of complex diseases and problems, you really need to have a multidisciplinary approach. And I think it's not coincidental that, as you can see here, we have PhDs and MDs working together. And to me, it's exhilarating. And I really can't think of anything more gratifying than to start with a human patient, identify a gene, and bring it back to now the family as one example that I'm going to introduce to you again, the Mitchells. So I will stop right there, and I think I will stop sharing my screen, and I'd like to introduce now Melissa Mitchell, who's going to share with you her very personal perspective of how our research has impacted their family.
Hi, I'm Melissa, and this is Cadence. Hello. And we just wanted to tell you a little bit about our story. We've been working with Dr. Grossfeld's for, well, you're 16 now, for 17 years now. And I first met him when I was pregnant with Cadence. I went into early labor and had a lot of tests done. And when I met Dr. Grossfeld, they explained what he thought was this mutation that maybe I carried. And I had open heart surgery when I was young, but back then, you know, you were fixed and kind of forget it. We thought we were just fixed and healed and nothing was a problem, but I had been experiencing a few more problems. It wasn't until I met Dr. Grossfeld that I really thought I should take a closer look at what was going on with me. So he diagnosed both kids with this gene mutation, and they had the pacemakers, as he spoke of. And then I actually went to the doctor, and I'll tell you the truth, I really kind of thought I'll just make him happy and go to the doctor, go see the electrophysiologist just to get everything checked out. But what I found was when I went there, they said, you have all these different kinds of arrhythmia happening and you need to have a pacemaker put in or you could die. And I thought, this is unreal. I would have never known about this unless I had met Dr. Grossfeld. And he explained his research to me, and he explained what they were learning. And really, I think I was kind of a guinea pig in a good way for them to see someone who had actually had this mutation and who could give a futuristic look at it. So I had my pacemaker put in, and about 3 months later, my heart stopped working on its own completely. So I was absolutely saved. And to this day, not a day goes by that we don't remember the moment that I was told your heart no longer beats on its own, you are surviving on your pacemaker, because I attribute my life being saved to Dr. Grossfeld and all of his research that he explained. That being said, my daughter, Cadence, has hypoplastic left heart syndrome, and she has an arrhythmia, and she had a pacemaker put in, and she's paced a lot. So you could see kind of a progression of her congenital heart disease, just like Dr. Grossfeld predicted with us based on his research. And then Chance was the same. He was born with the same issues as I was born with. And yet we didn't even know. And Dr. Grossfeld, took a look at him, and we did, again, more research and checked everything. And it all comes down to the power really of what research can do. We would have never known ever what our issue was. I thought I just had a hole in my heart and it was fixed, and I never would have dreamed something else would have happened to it. But he was very clear on his request for me to go get seen, and I'm so glad he did, because he definitely saved all 3 of our lives. That none of us would be alive today if we didn't have this powerful research. really figuring out what was wrong with our heart, what needed to be fixed. And I look forward to future treatments. I'm actually getting my pacemaker replaced in a couple months because the battery's running out. And it's just miraculous, the things that I've learned from him and the fact that we're all three alive thanks to him. I don't want to cry, but I'm very thankful to him.
Melissa and Cadence, we thank you so much. And I too am a survivor, and I am so grateful for the research that has saved my life. Thank you so much for sharing your story and explaining to us just in real life faces exactly how these types of collaborations and research save lives. So we're excited now to get to our audience questions. And I have some already in here, but if you're listening to this and you have more questions, go ahead and go to the bottom of your screen and you'll see where you can put in more questions. So the first question I have is for Dr. Grossfeld, which is, some of those interventions, are those going to be in vitro?
I don't think it's going to be feasible to go in and take the cells and replace the genetic defect itself, because by the time, even with more sophisticated imaging techniques that would allow us to even diagnose these defects very early on, in a way, genetically, the die is already cast. But as we learn about the functions of these genes, rather than fixing the DNA itself, what people might refer to as gene therapy, we hope to be able to restore the function of what those genes do to maintain, hopefully, normal cardiac development. That would be a best case scenario. But again, as I mentioned, in our frog model, we already have what appears to be a proof of concept that that might be feasible. Not to mention, of course, every gene is unique in its function. And we know, obviously, the pacemakers were done well after their hearts formed and well after they were born, but still obviously had a very major impact on their outcomes.
Thank you. I want to also mention that we have Dr. Karen Occurs with us here. She also works with Sanford Burnham, and she may be answering some of the questions. And I know she does wonderful work in this field, and we're delighted to have you join us here today. Thank you. So the next question I have, and you guys are going to decide who answers the question. So what tools, technologies would help accelerate the identification of CHD genes? Who gets that one?
Well, I can start and Paul can.
Thank you, Rob.
You can both answer that one, I think.
Well, one of the things is manpower. I think we have developed quite a bit of technologies how we can find many genes that have a function and test them in combinatorial fashion, sort of along the lines that I have been explaining. But it needs quite a bit of manpower and quite a bit of resources in order to do that. And then we would like to have also mammalian model systems then to test our best candidates and combination of candidates. So it comes down to money. I mean, simply put.
Great answer. Thank you. Okay, here's the second question. Are most CHD genes dominant, recessive, or do we know?
I can answer that very quickly. All the above.
Okay, so it's a smorgasbord. Okay, thank you.
I can add to that. I mean, when you see that the Tin Man had a weak mutation, didn't have an effect in the heart, so, but if you put it together with another weak mutation, now it becomes dominant. It was recessive before, right? So it all depends on the combination.
Okay, great, thank you. So would it be worthwhile developing potential testing for CHD?
Yeah, we already actually can do that to some extent, and really it comes down to cost. In theory, we could do whole genome sequencing, meaning sequencing all 23,000 genes of the fetus really very early on during the pregnancy. Up until now, the standard genetic test was just amniocentesis, but we can take that same source of material and apply state-of-the-art technology. So yes, it just comes down to money and whether or not insurance companies might want to support that or otherwise.
Wonderful, thank you. Good to know.
If I can chime in there.
Oh, please, Karen.
What we're finding out from our collaborations with the Mayo Clinic and what they know about the genetics of hypoplastic left heart syndrome is that there are hundreds and hundreds of variants, they call them, in every single patient. So the real challenge is to try to figure out which are the ones that are the most important to cause a particular disease and how they interact. And that's something that is a very big challenge for bioinformaticians and cannot be really reasonably done in complex model systems of humans. And so that's the basis for why we're doing what we're doing, is trying to figure out what the important connections are.
Can I tell you a little story on that? When we first started to collaborate with the Mayo Clinic, we looked at, several genes, several dozen genes, and for them, the hundreds and hundreds, the 1,000 genes that they had identified, it was going too slow. That would have taken a decade.
Yeah.
And so they gave us $2 million and we did it in 1 year, just to give you a perspective of how we do things.
That's thank you. All right. So if you discover a CHD gene, how could you go about fixing it in a baby, newborn, or in utero? I think we answered that question a little bit.
I think so.
Anybody have any to add to that, or we kind of answered that question?
Yeah, and just to follow up, I think it really more depends on what the function of that gene does, and that allows us to strategize our therapeutic approaches.
Okay. So are there any mammalian models? I think I got that right. Mammalian models for CDH? If so, would these add more insight into the disorder?
No, absolutely.
I don't understand the question, so.
Absolutely. No, there is mouse models, and people are developing also pig models and rabbits that are more similar to the human heart. And there's many mouse models of what one could call congenital heart disease. But there, the progress is just one gene at a time, maybe two genes at a time. It takes a lot of money and effort. So it's a matter of, do we want to first study the genetic networks and then the best candidates then have in the mouse models? And so this is basically our approach, to amplify the genetics, that then we can much more targeted look at combinations of 2 or 3 genes than in like a mouse model or so. But there are several models, yes.
So that goes back to your slide where you show that you have the human model and you had the different types of models come together, creating the.
Right, exactly.
Yeah. Okay, that was good.
And if I could just add to that, the mouse, my lab works on genetically engineered mice. And even with this one gene that we've discovered, depending upon what we refer to as the genetic backgrounds or the strains of mice, we can get some of these same congenital heart defects. However, when you're working with mice, for many reasons, it's extremely complex and time consuming. And I think one of the things that's been amazing to me, that to this day, we still have a hard time convincing grant reviewers, is the power of the fruit fly. And to me, I think it's incredible that these same genes that we've discovered and others have discovered to cause human congenital heart defects are actually conserved in the fruit fly. And so we can take advantage of a relatively simple, but still incredibly elegant system to study in a much more expedited and efficient way what these genes are doing, which then we can go back to the mammalian systems and ultimately humans. So complex.
And I would assume, though, that the fruit fly is a lot less expensive as a.
Absolutely.
Yes. I mean, just guessing here. Okay. So is bicuspid aorta a congenital defect, and what is the latest in treatment?
I can speak to that if you'd like. So bicuspid aortic valve typically refers to the appearance of an aortic valve. So if I were in the old days, I'd be up at a chalkboard and I'd draw for you like a Mercedes-Benz sign or peace sign. So normally the aortic valve has 3 leaflets that open independently. And in what's most people refer to as a functionally bicuspid aortic valve, 2 of the 3 leaflets are basically stuck together so that they function as one. So that's what gives rise to what's considered a bicuspid aortic valve. And remarkably, about 1% to 2% of the general population have a bicuspid aortic valve. And of those, about 15 to 20% of the time, they ultimately need some kind of intervention, surgery, or a catheter-based procedure to try to fix or repair that valve. So in terms of treatments, historically, the treatments are either a catheter-based procedure or sometimes open heart surgery to fix or even replace the valve. It's possible as we and others learn more about the genes involved in that process of what causes that abnormal valve, there may be some therapies on the horizon that will stabilize that bicuspid aortic valve so that everyone can be in that 80% subset that sure they have it, but it's of no clinical consequence.
Wonderful. Thank you. So did Melissa have a PFO or another procedure?
I guess, should I answer that, I suppose? Or
You
go ahead.
want to- you can, sure.
I think
So a PFO is actually a normal finding in about 20% of adults. It's just a little opening between the two upper chambers of the heart. So the PFO stands for a patent foramen ovale. Hers was much more extensive than that. It's called an atrial septal defect, where it was literally an anatomic defect that prevented the two atria from having separate circulations. And so in and of itself, over time, that can cause problems leading to premature death.
Okay, that's great. We have a question for Melissa here. So Melissa, are you aware of any family ancestors that you have suspected of having this problem? Is there anything in your ancestry.com?
Well, that's funny because when this all, she had attended the puppy, sorry. We had, when we met Dr. Grossfeld, we had done testing, you know, for my siblings and my mom. My dad had already passed away. So nobody else in my immediate family tested positive for this Tin Man gene. But I don't know enough information about the rest of our family. I have some history of acquired heart disease and heart attacks, and therefore that type of thing, but nothing as far as this exact problem. So
Thank you. Appreciate that. Okay, so I have another question here for one of the scientists. Can this hypoplastia also cause strokes? This is an interesting question, I'm wondering.
I can answer that. So the standard way that we take care of these patients is through a 3-stage surgical approach called the Norwood and Fontan. And I could spend hours really going into detail for answering that question, but yes, as a result of that Fontan procedure, there is potential risk for stroke in which actually blood clots form because of the intrinsic circulation of the Fontan anatomy that, if not looked after, could potentially lead to a stroke. Fortunately, thank goodness, in our patients, that's extremely rare because we're very proactive to use medical therapies and otherwise to prevent that scenario. But hypothetically, yes, it is possible.
Here's a question. How do I ask my cardiologist for this kind of genetic testing?
Maybe Melissa can answer that one since she has to deal with the insurance companies.
But you know what? It's super important. And a lot of my friends who have congenital heart disease try to find out how to get this kind of research, because I think that's just super important for treatment purposes and future, to really be proactive about it. It helped us quite a bit knowing, because we did go before we had children to have a genetic workup done to see if my heart problem was genetic. And that short amount of time before we had children, there was nothing. There was no evidence. And doctors said, you're fine. That's nothing genetic, and none of your kids are going to have that problem. So.
And thank you very much. So what I found going through cancer, that sometimes you just have to hound your insurance company to death. Yes. And eventually after 17 no's, they'll say yes. Yes. So here's another question. When a CHD is combined with another chromosome defect, are the interventions different than if the DX is only the Tinman defect? Is that?
Yeah, no, that makes sense. So I can start, and if anyone else wants to add to that. So maybe in about 20% of all cases of congenital heart disease, there are what we call extracardiac comorbidities. And so, it really depends upon the situation. The most common chromosomal abnormality associated with congenital heart defects is Down syndrome. In this syndrome that I study, Jacobson syndrome, because there are all these other adjacent genes that are also affected, there's a whole, range of potential problems that can go along with the congenital heart defect. So it just depends on the individual situation. Having said that, even one individual gene may affect multiple organ systems during development. So in certain situations, the one gene that might cause a heart defect potentially can lead to other problems like brain or kidneys or otherwise.
Great.
That is actually quite common. And so, for example, the Tin Man gene that is fairly specific to the heart, maybe then just one component that aggravates or causes the disease in the heart. But other genes, like in Notch and other transcription factors, they are actually working in many different tissues. And so combinations of these genes will aggravate heart defects, but also in many cases, actually neurodevelopmental defects have been associated with many of those gene combinations at this point. It is hard to prove that this is the defect until you can actually cure it in the model system. Like one question was whether one can cure this with CRISPR, and that can be done to some extent and could be a future sort of intervention to correct a defect. But if it's already an anatomical defect very early on in the embryo, that part is not easy doable. So then genetic testing is actually probably the key for future strategies of what to do about it.
You just answered one of the questions before I asked it. Yes, about CRISPR. Thank you. So I'm interested in statistics on families with hypoplastic left heart and bicuspid aortic valve, because my stepdad and 2 half-brothers died due to these 2 conditions. Can you speak to this correlation?
Yeah, there have been actually really good studies that show that if you look at first-degree family members of people with hypoplastic left heart syndrome, there is a much higher frequency of a bicuspid aortic valve and in general other heart defects. And so clearly that's one of the strongest arguments for the genetic basis. And what we would love to know someday is, why is it in the same family, for example, one person might just have a bicuspid aortic valve, whereas another child might have the full-blown hypoplastic left heart syndrome. So this speaks to, I think, what Ralph addressed before in that, and Karen, that there are really almost a roll of the dice to get the perfect storm combination of genetic factors that will manifest as these various different types of heart defects.
Yes, and one of the questions also was about, is this caused by environmental ever, or is it always genetic?
Now that's the $60,000 question, right?
Thanks.
we know that the environment that the fetus is developing in can perhaps play a role in terms of the hormonal milieu and things like that. But for sure, things that mom is eating and breathing will for sure have effects in
ingesting.
I can definitely speak to that. For hypoplastic left heart syndrome, there were some very good epidemiologic studies back from like the 70s that at least showed very strong correlations or associations. So for example, if both parents were, let's say, working in a factory exposed to organic solvents, their children had something like a 10-fold higher risk of having hypoplastic left heart syndrome. And there are sort of geographic clusterings of people were in towns, let's say close to chemical factories and whatnot, where there was a much higher than predicted frequency of babies born with hypoplastic left heart syndrome. So, at the end of the day, as we learn more about these genes and what they do, we're also going to learn how environmental factors might basically poison the function of these genes at just the critical time as the heart is developing. Yes. Another potential factor that's been put out there is a potential role for viruses. So it's, at the end of the day, it's as complex as cancer. I think you wouldn't have a very different answer, as you know, to that.
Yeah. If I can add to that, it's the genes, they don't function in the vacuum. They're in constant interaction with various environments, right? And the key thing of how this could influence gene functions or gene expression and so forth is epigenetics. So they are factors that are altered through epigenetic or environmental and other types of influences that can then, in many cases, aggravate or ameliorate the function and the disease of also the heart or other diseases as well. This is in constant interaction with one another. You can't separate the genetics from the environment really in how it affects you.
Okay, we have quite a few people here who have more questions and we're almost out of time. So my daughter is 14 years old and was born with HLHS. A second cousin of hers was also born with HLHS. Would we benefit from genetic testing for the future? Yes, we have all the panel.
Thank you.
So the answer to that
Please
is
email us.
I'm going to go to the next one.
I would love to actually study your family, because that could be extremely interesting if we could show common genetic variants between the two affected second cousins. That would be a wonderful opportunity to actually discover a new gene. So I very sincerely say, I hope you'll reach out to us after this. And
you got that?
We can follow up.
We'll be having that contact information so that you can do that. So
That's
wow,
actually the basis for the Mayo Clinic's collaboration that we did. They were examining parents and children and relatives, if they were available, with heart disease, and looking at the genetics within each family. And that's where we got the genes that we've been testing.
wonderful. Thank you, Dr. Kurz. That's very helpful. So I've heard some about Cardiac Atlas Project. Does that include genotype information in addition to phenotypic information? If not, is there something similar for cataloging? Anyway, you answered that part of the question.
Yes, we are working on that very, very heavily. And this is something. So the phenotypic information is observation. What is the defect? How does it look like? How does it manifest itself? How does it change over time? And all this, we know much less about the genetics and how to link it to what kind of manifestation, meaning phenotype, this then has. But that is exactly what we're trying to do, to figure out what combinations under what circumstances can give to what kind of manifestation of disease.
Okay, are there certain medications for certain types of CHD, or do they all require surgery?
Yeah, it depends on the circumstances. There are certainly medications that we use that can, relieve symptoms while we let sort of the natural history of the disease play itself out as to whether or not ultimately surgery is going to be required. And in other situations, all you need is medications, and then over time things get better. So it definitely depends upon the individual situation.
Okay, I've received the hook. I don't get to ask any more questions. So I want to thank our speakers, and we want to thank the Mitchell family for being here. I want to just remind you all that Sanford Burnham is specializing in these collaborations with doctors and researchers, and this is how things change and get better. And I'm so proud to be a small part of this organization. I want to thank you all who came and heard this presentation today. We will be sending out a recorded message for anyone who was registered. You're welcome to share that with other people. And we just thank you so much for being with us today and your dedication. And I am so proud of the scientists who presented for us today, and for your heart and your intelligence and your kindness. And I sit here because I've benefited from that, as the Mitchells have. So thank you very much. And I'm delighted that you were all able to join us today. See you again soon.